Hello, welcome to Biology, the scientific study of life. Repressible and inducible operons, two types of negative gene regulation. The trip operon is said to be a repressible operon because its transcription is usually on, but can be inhibited, repressed, when a specific small molecule, tryptophan, binds allosterically to a regulator protein. In contrast, inducible operon is usually off but can be stimulated induced. When a specific small molecule interacts with a regulatory protein, the classic example of an inducible operon is the lac operon, lac for lactose short term. The subject of Jacob and Monde's pioneering research, the described lactose milk sugar is available to E. coli. If the human host drinks milk, lactose metabolism begins with hydrolysis of the disaccharides into its component monosaccharides, glucose and galactose, a reaction catalyzed by the enzyme B-galactosides. Only a few molecules of this enzyme are present in an E. coli cell growing in the absence of lactose. But if lactose is added to the bacterium's environment, the number of beta-galactosides molecules in the cell increases 1,000-fold within about 15 minutes. The gene for B-galactosides is part of the lac operon, which includes two other genes coding for enzymes that function is lactose metabolism. The entire transcription unit is under the command of a single operator and promoter. The regulator gene LAC1, located outside the operon, codes for an allosteric to the operator. So far, this codes for an allosteric repressor protein that can switch off the lac operon by binding to the operator. So far, this sounds just like regulation of the trip operon, but there is one important difference. Recall that the trip repressor is inactive by itself and requires tryptophan as a co-repressor in order to bind to the operator. The lac repressor is, in contrast, is active all by itself, binding to the operator and switching the lac operon off. In this case, a specific small molecule called an inducer inactivates the repressor. For the lac operon, the inducer is allolactose, an isomer of lactose formed in small amounts from lactose that enters the cell. In the absence of lactose and hence allolactose, the lac repressor is in its active configuration, and the genes of the lac operon are silenced. If lactose is added to the cell's surroundings, allolactose binds to the lac repressor and alters its conformation, nullifying the repressor's ability to attach to the operator. Without bound repressor, the lac operon is transcribed into mRNA for the lactose utilizing enzymes. In the context, notice that of gene, this figure. The lac operon regulates synthesis of inducible enzymes. E. coli uses 3 enzymes, notice that, to take up and metabolize lactose. The genes are these 3 enzymes are clustered in the lac operon. The gene LACZ codes for B-galactosides, which hydrolyzes lactose to glucose and galactose. The second gene LACU codes for a premise, the membrane protein that transports lactose into the cell. The third gene LAC-A codes for an enzyme called transacetylase, whose function in lactose metabolism is still unclear. the gene for the lac repressor, LAC1, happens to be adjacent to the lac operon. An unusual situation, the function of the darker green, notice that, a region at the upstream left end of the operometer is revealed. Notice also lactose absent, repressor active, operon off. The lac repressor is notice that active, and in the absence of lactose it switches off the operon by binding to the operator. Notice the operator RNA made, no RNA made this. Notice also active repressor protein. Notice closer look, protein active repressor. Okay, in the context of gene regulation, the enzymes of the lactose pathway are referred to as inducible enzymes because their synthesis is induced by a chemical signal, allolactose in this case. Analogously, the enzymes for tryptophan synthesis are said to be repressible. Repressible enzymes generally function in anabolic pathways, which synthesizes essential and products from raw materials, precursors, by suspending production of an end product when it is already present in sufficient quantity. The cell can allocate its organic precursors and energy for other uses. In contrast, inducible enzymes usually function in catabolic pathways, which break down a nutrient to similar molecules by producing the appropriate enzymes only when the nutrients is available. The cell avoids wasting energy and precursors making proteins that are not needed. Regulation of both the TRIP and LAC operons involves the negative control of genes because the operons are switched off by the active form of the repressor protein. It may be easier to see this for the TRIP operon, but it is also true for the LAC operon. Allolactose induces enzyme synthesis not by acting directly on the genome, but by freeing the lac operon from the negative effect to the repressor. Gene regulation is said to be positive only when a regulatory protein interacts directly with the genome to switch transcription on. Let's look at an example of positive control of genes, again involving the lac operon. Positive gene regulation for the lactose utilizing enzymes to be synthesized in a appreciable quantity. It is not sufficient for lactose to be present in the bacterial cell. The other requirement is that the simple sugar glucose be in short supply, given a choice of substrates for glucolysis and other catabolic pathways. E. coli preferentially uses glucose. The enzymes for glucose breakdown glucolysis are continually present. How does the E. coli cell sense the glucose concentration, and how is this information relayed to the genome? Again, the mechanism depends on the interaction of an allostric regulatory protein with a small organic molecule. In this case, cyclic AMP, short-term CAMP, which accumulates when glucose is scarce, for 11.9 for the structure of CAM. The regulatory protein called catabolic activator protein, CAP, is an activator of transcription. When a CAMP binds to CAP, the regulatory protein CAP assumes its active shape and can bind to a specific site at the upstream end of the lac barometer. The attachment of CAP to the barometer directly stimulates gene expression, thus this mechanism qualifies as positive regulation. If the amount of glucose in the cell increases, the CAMP concentration falls, and without it, CAP detaches from the operon. Because CAP is inactive, transcription of the lag operon proceeds at only a low level, even in the presence of lactose. Notice this figure. Thus, the lag operon is under dual control, negative control by the lag repressor and positive control by CAP. The state of the lag repressor with or without bound allolactose determines whether or not transcription of the lag operons genes occurs at all. The state of CAP with or without bound CAMP controls the rate of transcription if the operon is repressor free. It is as though the operon has both an on-off switch and a volume control. In addition to the lag operon, CAP helps regulate several other operons that encode enzymes used in catabolic pathways. When glucose is plentiful and CAP is inactive, the synthesis of enzymes that catabolize compounds other than glucose generally slows down. The cell's ability to catabolize other compounds such as lactose enables a cell deprived of other of glucose to survive. The specific compounds represent at the moment determine which operons are switched on. These multiple contingency mechanisms switch an organism that cannot control what its host eats. Bacteria are remarkable in their ability to adapt over the long term by evolutionary changes in their genetic makeup, and over the short term by the control of gene expression in individual cells. Of course, the various control mechanisms are also evolutionary products that exist because they have been favored by natural selection. Notice also a positive control of the lag operon and catabolic activator protein CAP. RNB polymer has high affinity for the lag barometer only when a catabolite activator protein CAP is bound to DNA. Notice that RNA site at the upstream end of the barometer CAP attaches to its DNA site only when associated with cyclic AMP, whose CAP attaches to its DNA site only when associated with cyclic AMP. Notice that this figure. Notice that. Whose concentration in the cell rises when the glucose concentration falls. Thus, when glucose is present, even if lactose or also is available, the cell preferentially catabolizes glucose and does not make the lactose utilizing. Enzymes. Notice also lactose present glucose scarce. Notice that CAMP level high. Notice that active closer look, active CAP, inactive CAP, CAMP. Notice that RNA polymers can bind and transcribe. Operator. Notice here operator. These are represents barometer. LAC1, LAC-Z, notice that the stage here. Also inactive LAC repressor, notice that. If glucose is scarce, the high level of CAMP activates CAP, and the LAC operon produces large amounts of mRNA Notice that for the lactose pathway. Another B, notice that another figure, lactose present, glucose present, CAMP level low, little LAC mRNA synthesized when glucose is present, CAMP as scarce, and CAP here, notice that is unable to stimulate transcription. Notice that inactive CAP and RNA polymers cannot bind. Notice that operator and inactive lac repressor. Thanks for listening.